Hello everyone. In this video we are going to discuss about the structure of monosaccharides. So in the previous lecture we have seen about the classification of carbohydrates and the classification of monosaccharides based upon the number of carbon atoms. So here there are three different forms of monosaccharide when it comes to structures. So the structure of monosaccharides, they do have three kinds of structure, and the very first structure is the straight chain structure or open chain structure of the monosaccharide. So if you see the molecular formula C6H12O6, which is the glucose, and this is the structure of Fittig-Bayet straight chain structure. And among these three structures, the most importantly, Fisher's straight chain structure is most important monosaccharide structure. So if you see, this is the structure of the monosaccharide. You can observe 6 carbon atoms. This is the structure of glucose according to the Fisher's straight chain structure, and this structure is the most acceptable, and this is the structure which we are currently using in the biochemistry for the study of carbohydrates. So the Fisher's straight chain structure, or the straight chain structure proposed by the Fisher, that's why it is known as Fisher's projection formula. It explains the existence of 4 asymmetric carbon atoms at the position 2, 3, 4 and 5. That's why the glucose exists in 16 isomers. So due to the presence of 4 asymmetric carbon atoms at position number 2, 3, 4 and 5, if you substitute the symmetric carbon in the formula 2 to the power n, it will give 16 isomers. So the characteristic feature of the straight chain structure is the presence of a free aldehyde group. So the main important feature of a straight chain structure is the presence of a CHO group, as you can see in the structure of the glucose. So one, two, three, 4, let me draw this, 1, 2, 3, 4, 5 and 6. And as the carbohydrates are the polyhydroxy and keto groups, you need to arrange the hydrogen and hydroxyl group HOH, HOH, HOH. So the main difference is at the third position you can observe difference in configuration, where the hydroxyl group comes towards the left and hydrogen group comes towards the right. So this is the structure, this is which you can draw the structure of a glucose, which is the straight chain structure, and the molecular formula is C6H12 and O6. So these are the 6 carbon atoms, 1, 2, 3, 4, 5 and 6. And the second form of structure in monosaccharides is the pyranose ring. So the pyranose ring is an hexagonal ring, which means hexa stands for 6, right? So it is a 6-membered ring. It resembles the ring of a compound called pyran, hence called a pyranose ring. And the sugars containing pyranose ring are called pyranose sugars, and their glycosides are called as pyranosides. So if you can observe the structure here, so this is the pyranose ring. So this is the 6-membered ring. Six-membered ring, 5, 6. So if you want to derive this straight chain structure from this particular pyranose ring, then this will be the structure. You can see here in the presence of an oxygen atom in the ring, and these are some straight chain structure which you can call them as hemiacetal structures. So these are the hemiacetal structure of the monosaccharide, which is again one form of structure, hemiacetal rings. These are known as an hemiacetal rings. So how do the hemiacetal rings are formed? So hemiacetal rings are formed due to the condensation of hydroxyl group of the fifth carbon to the first carbon atom. So if you can see here, so let's draw the structure of the glucose. CHO, C, 2, 5 and 6. H, OH, H, H, OH and HOH. So this is the normal structure of the glucose. So if an acid hemiacetal form must be formed, then there will be a condensation of the hydroxyl group of the fifth carbon atom condenses with the first carbon atom, which leads to the formation of an hemiacetal ring like this. See, you can see here. So these are the hemiacetal ring structures, and this is the Fisher's projection formula for alpha D glucopyranose, and this is the beta D glucopyranose, where you can see the difference in case of alignment. At the first carbon here, HOH groups are differently aligned in case of alpha D glucopyranose, and in case of beta D the configuration alignment become reverse. So this condensation leads to the formation of a hemiacetal ring between the fifth and first carbon atom. And other form of structure of monosaccharide is the Haworth projection formula. So Haworth is an English chemist, derived the ring structure. The sugar molecule exists in two types actually. One is furanose and pyranose ring. About pyranose ring, which we have already discussed. So here the furanose ring is a 5-member ring structure which has a pentagonal shape. You can see in case of pyranose it is 6-member ring structure. It is a 5-member ring. As you can see the position 1, 2, 3, 4 and 5, it's a 5-member ring structure completely, and you can derive a open chain structure from this 5-member ring structure like this. And again it resembles a compound known as furan, hence it is called as a furanose ring. And the sugars containing furanose ring are known as furanoses, and the glycosides are known as furanosides. So we have seen an open chain structure of the carbohydrate, and the two ring structures which are furanose ring and pyranose ring, and the hemiacetal structure of the monosaccharide. So these are the forms of structures in case of monosaccharides. So let's discuss about the monosaccharides, some important monosaccharides. So the first comes the glucose, Here the glucose is a sugar and the molecular formula is C6H12O6. That is the most abundant monosaccharide and it is made up of water and CO2 during photosynthesis by plants and algae. So here glucose is one kind of monosaccharide, right? So during photosynthesis the carbon dioxide is actually reduced to form carbohydrates, and the carbohydrates in the form of the carbohydrates stored in the plant body in the form of starch, right? So the starch is a form of a carbohydrate. So that's why the statement is given like these are made up of water and CO2 during photosynthesis by plants and algae. So it is used by the plants to make cellulose. So the carbohydrates are used in the plant body in, to make cellulose, which is the component of the cell wall, and this is the most abundant carbohydrate in the world. This is the most important point. So even though we have n number of carbohydrates to discuss, here the most abundant form of carbohydrate is the cellulose. So it contributes for the composition of cell wall by all living organisms, as well as it contributes for the production of adenosine triphosphate, which is used by the cell as energy. So if you can notice that the energy is actually produced in the body by the oxidation of the glucose. So when the glucose gets, let me show you. So when the glucose gets oxidized, it will generate energy. So which means when we take food, the excess form of food is stored in the form of glucose or glycogen in animal body, right? So that glycogen will be broken down to glucose, and this glucose will get broken down to produce the energy. So here the energy, the excess form of energy will be stored in the form of adenosine triphosphate. So the carbohydrate, the glucose actually serve the purpose of the production of ATP, which is the energy currency of the cell. So this ATP will be used by the cell as energy. So whenever energy will be required by the body, this stored form of energy, which is ATP, will be broken down to generate energy and it will be consumed by the cell. So these are some structures. This is the skeletal formula for the D-glucose, and this is the Hayward projection formula for alpha D glucopyranose, which we have already discussed, and this is the Fisher projection formula of D-glucose, where you can observe the open chain structure here. So glucose for metabolism is stored as a polymer in plants, mainly as amylose and amylopectin, and in animals it is stored in the form of glycogen. So amylose and amylopectin, these are the polymers which are the stored forms in plant body, and if it in case of animals, the glucose is stored as glycogen, and the glycogen will be broken down into glucose, right? And the glucose is circulated in the blood of animals as blood sugar, and the naturally occurring form is the D-glucose. So do you know how the D and L glucose actually formed? So when you pass the plane polarized light towards a glucose medium, if it rotates the plane polarized light towards the left direction, then it will give L glucose. If it rotates the plane polarized light towards the right direction, it will produce a D glucose. So these are the isomers. These are the stereoisomers. So these are produced synthetically in comparatively small amounts and is less biologically active. So among this D and L glucose, so naturally occurring form is D-glucose, while its stereoisomer, which is L-glucose, is produced synthetically in comparatively small amount and is less biologically active when it compared to D-glucose. So glucose found in free state in fruits and other parts of the plants. In animals, it is released from the breakdown of glycogen in a process known as glycogenolysis. The process of breakdown of glycogen into glucose is known as glycogenolysis. So, as the D-glucose is the most abundant form and more biologically active form, we need to discuss about this. So, this is again a simple sugar and it's a monosaccharide, cannot be hydrolyzed further, which means it will not give subunits, some other kind of subunits when you hydrolyze it. So these are colorless. D-glucose is colorless and it's a crystalline solid, soluble in water, and it is sweet in taste. That's why it is called as a sugar. So the D-glucose present in free state in sweet fruits, specifically grapes, honey, etc. And it is present in the combined form in sucrose. In the combined form, it is present in sucrose, in cellulose, starch, glycogen. So it is called a fruit sugar, a corn sugar, and a grape sugar. So human blood contains 1 mg per ml of blood. If it exceeds, it leads to glucosuria, a condition where you can observe the presence of glucose in urine. So the human blood contains 1 mg per 100 ml of blood. If this concentration get exceed, it will lead to a condition known as glucosuria. So as I have mentioned earlier, the glucose upon oxidation provides immediate energy for cells. So it is described as the carbohydrate currency of the cell. So the glucose is regarded as the carbohydrate currency of the cell, because if there is no glucose, no oxidation will take place, and if there is no oxidation, no energy will be generated and stored in the animal body. So as it contains aldehyde group, it is known as an aldehexose. You can see the structures here. This is the Haber ring structure and it is the Fischer projection structure. Open chain structure and it's a ring structure. So D-glucose is optically active. It is dextrorotatory, which means the light will be rotated towards the right direction. So hence the name D-glucose, and it shows mutarotation, and it also exists in alpha and beta forms. So these are the isomers. They are called as anomers. So the alpha D-glucose and beta D-glucose are the isomers, and these together called as anomers, as they differ in configuration anomeric carbon atom 1. So the second monosaccharide is the fructose. So fructose, it is also known as levulose. This is the structural isomer of glucose. It is a ketohexose, which means the fructose consists of keto group in place of an aldehyde group. So in case of glucose, it consists of an aldehyde group. In fructose, the case is different. It consists of a keto group, which is known as C double bond O. And it is again a sugar. It is crystalline state, sweet and soluble. It occurs in plants and honey in the form of oligosaccharide and polysaccharide like inulin. So what is inulin? Inulin is basically a polysaccharide used by plants to store the carbohydrate. So it is most important found in roots or tubers. So it comprises fructose subunits and displays varying degree of polymerization. So the fructose actually stored in the form of, so fructose is a monosaccharide, right? So the fructose stored form will be the inulin. So inulin is a polysaccharide. So the repeated unit of fructose together composes the inulin. So So inulin is a polysaccharide and it's used by the plant to store carbohydrate, and it is basically found in roots or tubers. So inulin here is a soluble dietary fiber. It's a prebiotic used to boost digestive health, regulate blood sugar and weight loss, beneficial for bacterial growth in gut. Used in food manufacturing as a low calorie fat or sugar replace. It is a reducing sugar. It is again a reducing sugar. So reducing sugar basically consists of functional groups. Upon reactions will form the other compounds. So if it can reduce the Tollens reagent and Fenning's solution. It is a ketohexose and it is an optically active compound. It is levorotatory, which means it can rotate the plane polarized light towards the left direction. So that's why it is known as a levo sugar. The specific rotation will be the minus 92 degrees. The angle will be minus 92 degrees. So in the intestine and liver, which are the digestive organs, the fructose is converted into glucose, and only upon the conversion of fructose into glucose it will be used by the organs. So the fructose cannot be directly utilized by the organs, so the fructose must be converted into glucose, then the body organs will be able to use. So this is the structure of the fructose. So if you can see, there is no aldehyde group, only keto group are present in the structure of fructose, and it consists of CH2OH and CH2OH, the top and bottom, and here is the hydroxy and hydrogen group arrangement. So these are the alcoholic group, primary and secondary. So based upon this, anomers will be formed, anomeric rings will be formed. So this is one ring structure for the fructose. So the third one is ribose. So ribose is a simple sugar, again it's a carbohydrate with molecular formula C5H10 and O5. D-ribose is naturally occurring form, a component of ribonucleotides from which RNA is built. So RNA consists of ribose sugar, which is a ribonucleic acid, is built by the ribose, and so this compound is necessary for the coding and decoding, regulation and gene expression. So without ribose sugar there will be no RNA, and without RNA there will be no coding, or there will be no gene expression. Like, so the RNA do consists of codons on it, right? So if you study transcription and translation, so this is the messenger RNA, and the messenger RNA do consists of codons on it. So if you start with AUG, which is the initiator codon, let me draw here. So this is the mRNA, which is made up of ribose sugar. So this mRNA consists of codons AUG, UUU, and some other kind of codons which codes for a specific protein. So for example, UU stands for codes for amino acid phenylalanine. So this kind of amino acid along with phenylalanine will form a protein molecule. So until unless mRNA composition is perfect, there will be no proper codon reading. So that's why the ribose sugar is utmost necessary for the composition of RNA. So if the RNA is stable and proper enough, then it will provide the codon, and upon good proofreading of the codon, there will be the formation of protein. Okay, so these are some structures of the ribose sugar. These are the ring structures, pyranose and pyranose ring structures of the ribose. And this is the isomeric form of the ribose sugar, which is D-ribose and L-ribose sugar. can see the difference in alignment. So these are the mirror images, which are the stereoisomers, which are mirror images to each other. So ribose, as it's a 5-phosphate ester, is typically produced from glucose by the pentose phosphate pathway. So the ribose actually formed by the pathway known as a pentose phosphate pathway. So in at least some RK and alternative pathways have been identified. So mostly the ribose is produced from glucose by a pathway known as a pentose phosphate pathway. So if it in case of RK, some other alternative pathways have been identified apart from pentose phosphate pathway. So here the ribose can be synthesized chemically, but commercial production relies on fermentation of glucose using genetically modified strains of Bacillus subtilis. 90 gram per liter of ribose can be produced from 200 grams of glucose. The conversion entails the intermediacy of gluconate and ribulose. So here, if you want to produce ribose sugar commercially, you have to start the process from the fermentation of glucose only, because naturally also the glucose can be produced from the should be produced from the ribose should be produced from the glucose through a pathway known as pentose phosphate pathway. So commercially also, if you want to produce ribose sugar, you have to start the process with the fermentation of the glucose. So here the glucose will be the raw material for the production of the ribose sugar synthetically and commercially. So for example, so this process can be started with the fermentation of glucose, and you have to use a genetically modified strains of Bacillus subtilis. They have given some example in which if you want to produce 90 gram per liter of ribose, you have to ferment 200 grams of glucose. So in this process, some intermediate products can be formed, which are known as gluconate and ribulose. So this is the process. So here the ribose is an aldopentose, which means consists of an aldehyde group. In the conventional numbering scheme for monosaccharides, the C atoms are numbered from C1 to C5. So the ribose consists of 5 carbon atoms. That's why the molecular formula is C5H10 and O5. ATP is derived from ribose. It contains 1 ribose, 3 phosphate groups and an adenine base. ATP is created during cellular respiration from adenosine diphosphate. So here the ATP is derived from the ribose. If you see the structure, the ATP do consists of 1 ribose sugar and 3 phosphates and an adenine base, adenosine triphosphate, right? So the ATP is produced during cellular respiration. How do the cellular respiration occurs? As I have mentioned, the stored form of glucose in the tissues gets oxidized to produce energy in the form of ATP. So upon cellular respiration, the ADP will be converted into ATP upon the addition of 1 phosphate to the structure. So ribose is a building block in secondary signaling molecules such as cyclic adenosine monophosphate, which is derived from the ATP. So the CAMP, which is a secondary messenger for the signaling pathways like translation and transcription. So these are the signaling molecules which actually contributes for the triggering or mechanism of some cell signaling pathways, which are utmost important for the cellular metabolic mechanisms. So D-ribose is a structural backbone of the purines and pyrimidines. Again, it is a critical cellular compound which are components of genetic material, as RNA is act as genetic material in some organisms like viruses. So this is the one utmost important critical cellular compound, and it is a constant of numerous cofactors and certain vitamins and ATP, which is the energy currency of the cell. So the D-ribose can be obtained from the diet, especially from foods containing high amount of riboflavin, which is vitamin B2, which is contributes for the metabolism and energy production. So in dietary factors, the ribo we have to one has to consume foods which are rich in vitamin B2, which contributes the for the composition of ribose sugar in the body, and it can be absorbed into the bloodstream from the oral cavity and intestinal tract. So the intake of riboflavin gets absorbed into the bloodstream from the oral cavity intestinal tract. So the levels of D ribose in the blood and cerebrospinal fluid are 0.01 to 0.1 millimolar in healthy individuals. So here the most important point, the D-ribose sugar also contributes for the composition of cerebrospinal fluid, is 0.01 to 0.1 millimolar in healthy individuals. So what is cerebrospinal fluid? So cerebrospinal fluid is the fluid which actually found in the regions of brain and spinal cord, which actually protects the central nervous system from the mechanical shocks and damages. So in this cerebrospinal fluid, the composition of D-ribose can be noticed, which is 0.01 to 0.1 millimolar in case of healthy individuals. And free D-ribose can also appear as an intermediate in nucleotide metabolism. So, these are the structures of monosaccharides, different structures of monosaccharides, and we have seen the examples for the monosaccharides, which are the glucose, fructose and ribose, which are at most important along with structures. I hope you understand this lecture. Thank you.